Hello students, welcome to my channel. We are discussing important questions of chapter number 11 and we are on video number 5 for this chapter. It is asking, the first question is, plasmid is extra chromosomal DNA. So other than the chromosomal DNA, bacteria also has extra chromosomal DNA in the form of plasmid. A piece of gene which is used to hybridize with the vector is gene of interest. Overhangs are produced by restriction enzymes. They are known as sticky ends. Transfer of DNA into the bacteria by phage is known as transformation. X-gal will be converted to a colorful product only when beta-galactosidase would be there. The machine that is being used in the PCR is a thermocycler. Thermostable DNA polymerase was being taken from Thermus aquaticus. Before integrating DNA into the bacterial plasmid, bacterial cells are treated with calcium. Why? Now bacterial cells are being treated by calcium to make them as the competent one, because DNA is hydrophilic in nature and it cannot penetrate like this. They are being treated by calcium to make them competent, and this helps in DNA insertion into a cell. You have also read about the heat shock treatment, that high temperature, low temperature, then high temperature is again being given, right? So this is the answer to question number 8. Now we'll move on to next one, and it is asking us, how many types of restriction endonucleases are known? So we know type 1, type 2 and type 3 endonucleases. Why are they being used as, or they being called as molecular scissors? So by taking one of the example of EcoR1, you can explain that if we have a DNA sequence with a palindromic sequence in between, then EcoR1 will recognize that sequence and will break the DNA. Because it is breaking the DNA, that's why it is acting as a molecular scissor. So you can also explain this by taking the example of EcoR1, SMA1 or any example you want to cite. Next question is question 10. Explain the principle of insertional inactivation by giving a suitable example. So for this I'll be sharing a link in the I button. You can check on that. Same is the insertional inactivation question number 11. So same video, same I button will help you for solving both questions 10 and 11. Why type 2 restriction endonucleases are used in recombinant DNA technology? Because they are easy to use and their recognition sequence is 6 nucleotides long, which is easily being helpful in breaking the DNA into parts. Here again you should cite one example of EcoR1 while writing this question. Question number 13 is again on PCR, and it is asking us, name the source organism that processes dark polymerase. What is so special about the function of this enzyme? So Thermus aquaticus is the source organism. Speciality is this, that it is thermophilic in nature. It can bear the high temperature without getting denatured, right? So if such question comes, then it will be generally for 2 marks. Please do answer each part separately and clearly. Next question is question number 14. They have given you an image and they're asking what is happening. Human DNA is seen, isolated plasmid is being seen, and you can see that gene of interest is being ligated to the plasmid to form recombinant DNA, right? So name the particular technique. So this is recombinant DNA technology which is being visible, and you can explain it in the form of steps, like first the isolation of gene of interest, treating them with the restriction endonucleases, and then ligating them together to form the recombinant DNA plasmid. So these were few of the important interesting questions from chapter number 11. However, I will definitely suggest that go through the NCERT, go through the complete video tutorial of this chapter. Once you go to my channel, just go in the playlist. In the playlist, there are different chapters, so you can read the chapter of your own choice. Please do not forget to share my channel with your friends and family if there is any in the family member. It will definitely be helpful. And in case of any problem, you can leave your problem in the comment section or you can email it to me. Thank you.